So one of my research focus is studying the immune microenvironment. The immune microenvironment consists of the immune cells and the tumor cells in the bone marrow and blood of patients with multiple myeloma. And we use a sophisticated technology to interrogate the bone marrow microenvironment. It's called single-cell sequencing. And so we can do gene expression profiling of every single cell in the sample. And that allows us to, one, characterize that cell, determine what kind it is. Is this a T cell? Is this a natural killer cell? Is this a myeloma cell? And then we can get a sense as to what that cell is doing. Is that cell activated and likely to kill a tumor cell? Or is it quiescent and just really not going to do anything? Once we can categorize and type all the different kinds of cells inside of the immune microenvironment, we can compare different time periods. We can say, okay, this patient's immune system profile looked like this before treatment, but then it changed to that after treatment. Furthermore, we can do it across a population, and we can say, I see that patients whom get this same treatment have a change, let's say, in the natural killer cells consistently over time. But maybe there was a subgroup that didn't, and that group did not get a good outcome. So we can find those kinds of patterns in the immune system across time, across patients, to be able to learn about why the treatment's failing or why the treatment works, and deciding which patients may benefit most from some kind of immune therapy versus another. We're also using this technology to be able to study myeloma at its earliest phase, in the MGUS, monoclonal gammopathy of undetermined significance, or smoldering myeloma phase. These are those two pre-cancerous states where there's a chance they could become myeloma, but they may never. And we can look at the immune cells in those cases and try and figure out what is it that makes a patient with these precursor conditions more likely to develop myeloma versus not. Most of the technology has been focused on looking only at the tumor cells to answer that question. But we believe the immune system also plays a really important role. We think the immune system may enable some cancers to grow faster than others, or some immune systems are stronger and prevent it from happening altogether. So those are the kinds of questions that we're really focused on in the lab at this time.
How long does single-cell sequencing take?
We have really cool technology. That's one of the things I love about being a physician scientist, is just the tech I get to play with. So one of these is a machine that actually can suck up cells in through a microfluidic circuit and then sort them into little oil droplets. And then in each oil droplet, it does a chemical reaction that allows it to, we call it barcode, each of the individual molecules inside of the cell, and barcodes the cell itself. So then when we release all the oil droplets, all of the molecules have a tag that say what cell they came from and what kind of molecule they are. And then we can sequence those molecules and then use computer software to be able to put it all back together again. So it happens in a period of hours to do these kinds of experiments. But to do it on lots of patients requires months, because it takes a long time to process one sample at a time.
Is it possible to know where cells are in relationship to each other?
Bone marrow is solid, and bone marrow is where myeloma mostly hangs out. And so we take cores of the bone marrow, and then we can slice them, and then we can put those on slides. And there is a technology we're experimenting with called spatial transcriptomics, where you can determine the expression profile of each cell in that biopsy. And so now you not only know the expression of that single cell, but what's its positional relationship to all the other cells. So it's like having an address to where it came from. And then once you know all of its positional relationship, you can say, are there certain cell types that like to hang out with each other? And if they are hanging out with each other, they might be communicating. And if they're communicating, could they be sending growth signals to each other or telling the other cell to die? And so those are the kinds of questions we try and tease out using spatial transcriptomics.